And the session is focusing on drug resistance. And drug resistance is all about the reduced effectiveness or ineffectiveness, in fact, of medicines such as antimicrobials or antineoplastic agents in treating disease or conditions. You know, this becomes a huge clinical challenge when trying to control disease. And without further ado, I don't want to steal the thunder from the experts who speak about this very eloquently. I would like to introduce our panel, and I will introduce them in turn. The panel of speakers that we have includes Professor Paul Race, and I will introduce him just now, Professor Greta Dreyer, and Dr. Deepak Timiri Govindaraj, who is our own colleague from the head of our Synthetic Biology Center. So, Professor Race, you are leading the conversation. Allow me to introduce you ever so briefly. I did try to summarize your bio, but it's still very long. So I'll just say some of the things, but not all the things. So Prof. Paul Race is a professor of biological chemistry and a former Royal Society University Research Fellow, based within the School of Biochemistry at the University of Bristol in the United Kingdom. His research focuses on exploitation and manipulation of natural product biosynthetic pathways for the delivery of functionally optimized non-natural product pharmaceutical leads. That's a very interesting nuance there. He is also the founding director of the Bristol Biodesign Institute and is the Bristol lead for the Engineering and Physical Sciences Research Council in the UK, funded by the UK Innovation and Knowledge Centre in Synthetic Biology. He's been co-director as well of many, many important things, which if I were to read out, that would pretty much take up his whole presentation slot. So it's between me reading his CV and letting him speak. And I choose to let him speak. So Professor Race, please speak to us. Thank you.
Thank you, and thanks for the very generous introduction as well. So firstly, I'd just like to thank the organisers for the invitation and for the opportunity to speak to you today about some of the work that's currently taking place in the lab here in Bristol. I'd also like to apologise for unfortunately not being able to be there in person, but unfortunately my teaching schedule dictates that I'm not allowed to travel away from Bristol this week. What I'm going to do over the course of the next 20 minutes or so is just give you some examples of work that's currently taking place in the lab here in Bristol, which is focused on the application of synthetic biology for the development of solutions to the emergence of antimicrobial resistance. And in the context of this talk, at least, I'm going to be referring to synthetic biology as the application of the principles and practices of engineering to biological systems. So, as I'm sure that all of you will be aware, we currently live in an era where we're seeing an increasing degradation in our capacity to treat infectious disease. And one of the primary drivers for this is the emergence of antimicrobial resistance, so the capacity of microorganisms, and in particular bacteria, to circumvent the effects of antibiotic agents that we use in the clinic. So in this slide, I'm just showing you on the left-hand side the list of chemical classes that we currently have available to us for use as antibiotics in the clinic. So here we have the 35 classes of antibiotics. And the orange bars on this plot indicate what we classify as the development window. So this is the period of time that takes place between when these individual groups of molecules were discovered in the laboratory to when they transitioned to clinical use. And the blue bars, these indicate what we refer to as a resistance window. So this is the period of time between which those molecules made it into clinical usage and there was the first reported emergence of strains of bacteria that could withstand the effects of those antibiotics. And if we take all this data together, this really two trends start to emerge. So the first of those is that over the past few decades, we've become much less good at discovering new antibiotics. And that trend is further reinforced by the data that's shown in this plot here, where you can clearly see that really since the 1980s, there have been very few new classes of antibiotics that have been discovered. And the second trend that emerges from this data is the fact that many of these classes of antimicrobial drugs are now becoming increasingly less effective because there are strains, clinically important strains, which are now resistant to the action of these drugs. So what this means together is that there's now a real pressing requirement for us as a global community to start to address this issue of reactivating or re-energizing the antibiotic discovery pipeline. Now, historically, our favoured source of antimicrobials or antibiotics were natural products, so molecules that are sourced from natural sources like other microorganisms or plants. And in fact, around 70% of all the antibiotics that we use in the clinic today are based on or derived from natural product scaffolds. And that's even more astonishing when we consider that in the 1980s, the pharmaceutical industry really made a decision to turn their back on natural product drug discovery. And it's kind of against this backdrop now, with the view that that was probably a mistake, that there is renewed enthusiasm for researchers to turn their focus back to natural products as the starting point of new antibiotics. So how do we address that in the lab in Bristol? Well, really we take a kind of three-pronged approach to solving this problem. So firstly, we operate a biodiscovery program out of the lab where we go and we try to identify and isolate strange and unusual microorganisms that reside in environmental niches that have never previously been explored, for example, the very bottom of the ocean. And then we try and identify from those strange and unusual microorganisms the bioactive scaffolds that those microorganisms produce that could form the starting point for new antibiotics. In parallel, we operate a stream focused on both understanding at the molecular level how those molecules are produced within those microorganisms, so the enzymology and the biochemistry that's involved. And once we have that fundamental knowledge, we can then use that information to try to manipulate or modify those pathways to give us access to functionally improved variants of those molecules. And these are molecules that we refer to as non-natural natural products. So they're much more viable targets for development as a clinically useful antibiotic. And in doing this work, we also frequently identify novel biocatalysts or enzymes from these pathways that can be used and applied out of their natural context to build, if you like, de novo chemical scaffolds, which again, that can be taken forward for antibiotic discovery and development. And tying together these three strands of activity is an ambition to really take the outputs of this work and turn it into something which is clinically useful. And we do quite a lot of work in collaboration with partners in industry, both in the UK and also internationally. So this slide just provides a little bit of a summary of some of the exciting targets that we've been working on in the laboratory over the past few years, maybe the past 5 or 6 years. And what I'm going to do for the remainder of this talk is just show you 3 examples fairly quickly of how we've been using synthetic biology-based approaches to try and unlock and realize the potential of some of these natural product scaffolds. And I'm going to show you 2 examples that involve using in vivo tools, so actually engineering pathways in producing microorganisms. And then the last example that I'll show you is using a more in vitro-driven approach, where we reconstitute part of the biosynthetic pathway in a test tube in the laboratory. And all of these approaches are really underpinned by this philosophy of using synthetic biology. So the first example that I'm going to tell you a little bit about relates to a clinically used antibiotic called mupirocin. And the predominant active component of mupirocin is this molecule here. This is pseudomonic acid A. Mupirocin is produced by this bacterium called Pseudomonas fluorescens, which is a soil-dwelling bacterium, and it's sold commercially. It's produced by GlaxoSmithKline as the active ingredient in this topical cream called Bactroban, and this is used extensively in the treatment of skin infections in burns victims or people who've undergone skin grafts. So mupirocin has a very encouraging spectrum of activity, but due to its chemistry, there is a limitation on its application. So it can only be used as a topical application for reasons which are illustrated here. So pseudomonic acid contains this 10,11 epoxide group, and if you acidify this molecule, you can get a tack at either face of this epoxide group to generate these two cyclized products. Now, the way that mupirocin functions as an antibiotic is it inhibits a key enzyme called isoleucyl tRNA synthetase. And in fact, if you generate these two cyclized products, neither of these can effectively bind to the target site on the isoleucyl tRNA synthetase. And for that reason, mupirocin is not tractable for use as an antibiotic for systemic administration. It can only be used as a topical agent. However, there is some good news. It's not all bad news. So if we look at the composition of mupirocin, it's actually made up of three different components. So the predominant component is this pseudomonic acid A, which contains this 10,11 epoxide, and this is a fairly effective antibiotic. There's also a minor component in mupirocin, which is this compound, pseudomonic acid B, differs through possession of this hydroxyl group. Now, pseudomonic acid B isn't in its own right an effective antibiotic, so we can discount that compound. But there's a second minor component of mupirocin, which is this compound here, pseudomonic acid C. And what's particularly exciting about pseudomonic acid C is that not only is it at least as good an antibiotic as pseudomonic acid A, but it lacks this 10,11 epoxide group. So in principle, this compound will not be subject to the same deleterious cyclization effects that we see in pseudomonic acid A due to this epoxide. So this could be an attractive target for taking forward as a novel antibiotic. So what we really set out to do was to use synthetic biology to see if we can engineer the mupirocin pathway such that we can generate a strain of Pseudomonas fluorescens which only produces PAC and produces this compound at a level that makes manufacturability feasible. So I'm not going to go through all this chemistry in intricate detail, but basically to achieve that outcome, what we had to do was, firstly, we had to establish the molecular details of how pseudomonic acid C and pseudomonic acid A are biosynthesized in the mupirocin pathway, and we were able to resolve the individual steps and enzymes involved to do that. But what I want to do is really focus your attention here on this intermediate in the biosynthesis of mupirocin, because this is the stopping off point for the generation of pseudomonic acid C, which is the compound that we'd like to make access to. Now, an obvious approach to ensure that we get pseudomonic acid C exclusively from our strain is simply to knock out or generate a mutation in the epoxidase, which is this enzyme MMP-EOR. And of course, we did that at the kind of conception of the project. But unfortunately, in that single mutant, what we find is that it does produce pseudomonic acid C exclusively, but unfortunately, it produces at incredibly low titers. So we need to get around this problem, and actually what we discovered through a mutant screening process is that if we combine this mutation here with mutations in 3 other enzymes that operate downstream within the mupirocin pathway, we're able to produce a strain that makes exclusively pseudomonic acid C and at a level that makes manufacturability via fermentation a tractable alternative. And that's just illustrated here on this slide. So here we have an extract from the wild-type Pseudomonas fluorescens strain, and you can clearly see that the predominant species here is pseudomonic acid A, with vanishingly small amounts of B and C, whereas in our engineered strain, this strain produces predominantly pseudomonic acid C in an incredibly high titer, and this now has been taken forward through our existing collaboration with GSK to see if we can generate a systemically deliverable variant of mupirocin that circumvents the existing issues with the pseudomonic acid A-based treatment which is currently used. So that's example 1, and I think that's a nice illustrative example of using synthetic biology tools in vivo to kind of solve this problem. The second example I'm going to talk to you a little bit about concerns another bacterial strain. This time it's a Micromonospora strain, and this was a strain that we identified as part of our marine biodiscovery program. So this is a strain that we isolated from the microbiome of a deep sea sponge. And the reason that we became interested in this microorganism was due to its capacity to produce these 2 compounds here. So this is a molecule called casinostatin, and this is its isomer isoquinoline B. And the reason that we were interested in these molecules is that they display an encouraging spectrum of activity as antimicrobials, in particular against some high priority microbial strains. So Acetobacter baumannii and also multi-drug resistant strains of Staph aureus, so various different flavors of MRSA. So what we wanted to do was explore the feasibility of taking these compounds forward as prospective new antibiotics. But there was a catch, and that catch was that the authentic natural producer, the Micromonospora strain that we secured from the bottom of the ocean, produced very low titers or very low quantities of these individual compounds. So what we set out to do was to try and to make engineered variants of those strains which are able to produce these attractive molecules in much higher quantity. And the way that we did that was to use an approach referred to as ribosome engineering. So what you do in ribosome engineering is you take your parental strain and you grow that strain in the presence of an antibiotic that selectively targets the ribosome. Okay? And then you then select for mutants which are resistant to that antibiotic. And that resistance must emerge due to the acquisition of mutations within the ribosome of that strain. Now, nobody really understands explicitly why this happens, but it's been shown reproducibly by many groups that the acquisition of resistance mutations within the ribosome promotes and upregulates the expression of genes involved in natural product biosynthesis. So what we did in this experiment is we took our marine strain of Micromonospora, we grew it in the presence of increasing concentrations of rifampicin, and then we selected across multiple generations for a hyperproducer of casinostatin. And this really summarizes the outcome of that experiment. So in the top left-hand corner, this is our natural strain that we isolated from the bottom of the ocean, and it's producing casinostatin, and you can see that through this zone of inhibition around the strain. And then here we have generations 1, 2, and 3 of our mutants that we've generated through screening in the presence of rifampicin, and you can clearly see that the zone of inhibition for this strain here is much larger than what we see with the starting strain, and that's because this strain has acquired mutations as part of the screening process in the ribosome that upregulates expression of this gene cluster. And in fact, we've sequenced the genome of this mutant, and we can relate that upregulation in expression of that gene cluster to a single amino acid substitution within the ribosome. So small change that makes a huge difference. Okay, so that's another example of using an in vivo synthetic biology approach. The last example I'm just going to quickly show you relates to adopting a more in vitro centric approach, and it focus is on this antimicrobial compound here called abyssamycin C. This is a spirotetronate natural product antibiotic, which is a potent inhibitor of a key enzyme involved in bacterial folate biosynthesis. So this is a very attractive target as a starting point for a new antibiotic. What I want to draw your attention to specifically in the biosynthesis of this molecule is this step, which is illustrated here. So it's a reaction which is catalyzed by an enzyme called ABYU. And what's unique or special about this reaction is the fact that ABYU was shown to be the first ever confirmed naturally evolved Diels-Alder biocatalyst. So ABYU is a Diels-Alderase, and the reaction that it catalyzes is a 4+2 cycloaddition between this exomethylene group in this pathway intermediate here and a diene within the triene of this portion of the molecule. So 4+2 cycloaddition that brings together this diene and this dienophyl to form this heterobicyclic spiro system. So what we were able to do in the lab was produce ABYU in recombinant form and then synthesize a derivative of the authentic substrate for this enzyme and confirm using liquid chromatography coupled to mass spectrometry and NMR analysis that this enzyme did indeed catalyze this 4+2 cycloaddition reaction. So that's a fundamental discovery. What we then wanted to do was explore the possibility of using this enzyme to generate alternative derivatives of this intermediate that we could then take forward and convert to non-natural abyssamycins. And this slide just shows you some examples of how we were able to do that. So at the top here, this is the natural substrate with the wild type enzyme that makes this intermediate here. But we're also able to invert the stereochemistry at this position, or move the location of that methyl group, or remove any kind of methyl decoration whatsoever. And in fact, the enzyme has a sufficiently broad substrate selectivity to act on each of these derivative substrates and generate these products. So these three products here are molecules which have never been observed in nature. They are genuinely unnatural products. So that's fine as a proof of concept, and I'm going to finish off with this slide. But what's really exciting here is that we can also start to deploy this approach to generate molecules that may have increasing potency as opposed to obisomycin C. So in this example, we've taken a similar approach. We've synthesized a variant of the natural substrate, but in this instance, installing an additional carbon, so extending the length of that chain. And we've been able to show in vitro that if you incubate this substrate with our enzyme ABYU, we can generate a new product, which equates to this molecule here, albeit the enzyme accepts this substrate less well, but it's still able to act on it to produce a molecule that has an extended macrocycle. And we can complete the synthesis or biosynthesis of this molecule to convert it into an abisomycin-like compound and screen its activity. And that's what you're looking at here. And what we can clearly see is this expanded macrocycle variant displays increased potency in an agar plate-based screening approach as compared to the authentic natural product. So in this example, we've been able to use an in vitro rather than an in vivo synthetic biology approach to generate a functionally optimized variant of that natural product. So what I'm going to do is I'm going to conclude there. Just to summarize, hopefully what I've been able to do is convince you of the power of synthetic biology and how it can be used and deployed to produce improved variants of existing antibiotics. And I'll just conclude by thanking all the individuals that have contributed to this work, and also thanking our funders who are listed over here. And thank you for your attention.
Thank you very much. And so it's quite intriguing to me that we see a flattening of our rate of discovery, like you said, and yet we've got very potent tools for discovering and tweaking and carrying on. So one would think we should be seeing an explosion of new products coming onto the market, given the powerful tools that we have. But kindly hold the thought, because I would like you to address it during our Q&A session when we are done with the rest of the presentations. Thank you very much.